Hello, everyone. My name is Manoj. I'm a PhD candidate at the University of Sydney and Children's Medical Research Institute. So today I'm going to be talking about an interpretable deep generative framework, which I developed for capturing cell identity paired with an automated annotation tool for single-cell multi-omics. So, as you might know, compared to bulk sequencing, single cell sequencing is able to capture the molecular variability occurring in each individual cell, thereby providing a high resolution view of the cellular heterogeneity. However, analyzing single cell data is not an easy task because single cell data is often high dimensional due to a lot of genes, for each cell. It's often noisy, there is sparsity and dropout events. So analyzing this data requires development of robust methods. Although statistical and classical machine learning based methods have made effort in analyzing this data, recently deep learning has gained increasing popularity in analyzing this data. However, deep learning models lack interpretability and obscure the reasoning behind their predictions. So to address this, we developed an interpretable framework, which we call Hydra. So here I have simplified the framework, but the basic idea is we train a variation encoder in a specialized manner and do multitask learning from the single cell omics data, which can be individual or those generated by joint profiling. Once we do this, we generate a balanced data set by randomly downsampling the major and producing new minor samples and fine-tuning on these balanced data sets multiple times to capture the cell identity genes. Finally, we use this to perform an automated cell type annotation using an ensemble of simple neural network classifiers to annotate the single cell data. So first thing I want to talk about in terms of results is the stability of the features. Uh, so what I mean by stability of features is being able to select consistent set of features, which means a small variability in the data set should not cause your model to select completely different set of features or cell identity genes. So that's why feature stability is quite important. Here we compute the feature stability by using Pearson's correlation coefficient. I'm comparing against four very well-known statistical methods, lemma test, t-test, HR, and Wilcoxon. Specifically, I'm comparing this because previously we observed that these methods often have quite high reproducibility compared to a deep learning model, but our hypothesis was ensemble can improve this. And as you can see, we are performing very well in terms of feature stability. So I've done using two long data sets to compute this. Next is about selecting cell identity genes. So one of the good things about our framework is that it's able to effectively capture cell identity genes. So what I mean by that is here I'm taking the mouse cell atlas and a specific cell type of interest, that is the kidney PCT epithelial cell. So basically cell type specific genes, for example, the gene selected for this particular cell type should show expression specifically in this cell type. And that's exactly what we are observing here. As you can see, the red color represents the log normalized expression, and these are the top genes provided by our framework, and it's showing expression specifically in our cell type of interest. And then I compare whether this expression is specific compared to all other cell types and how plotted a ridgeline plot. And that's exactly what we are observing, which means our model is able to capture cell type specific genes, which can be useful in identifying cell types. Next, I want to talk about annotating cell types from single cell transcriptomic data. So first, I'm showing you an intra-dataset cell type annotation from a processed urethra dataset. So what I mean by intra-dataset cell type annotation is I'll be splitting the dataset into train and test, and training on train data and testing on test. So this is intra-dataset cell type annotation. For a quick evaluation of the model, here all cell types are arranged from smallest to largest, and this is the overall. As you can see here, these cell types perform quite well for major cell types, the other methods, but they often suffer when it comes to minor cell types. That is mainly due to dominance. However, we are able to keep this consistent performance across almost all of the cell types And overall performance is of our method is quite better compared to these very well-known methods. Next, what we have is the inter-dataset cell type annotation, which means I'll be comparing across batches, evaluating across batches, or generated by different experiments, different technologies, or from different labs. So here I'm evaluating on 25 trained test pairs across 4 different tissues: kidney, lung, PBMC, and retina. And the methods have been ranked based on their overall accuracy. And our method seems to perform quite well compared to these other methods. And I'll also include a rank to show the individual dataset performance as well. Next, what we have is annotating cell types from single-cell multimodal data. So here I've included the other one in the poster, but here one of the datasets I wanted to show you is NARSIC, which is RNA plus ATAC on a brain dataset. And I'm comparing against recently published methods for multimodal data, that is MOFA plus, UMIN, and SMOMAT. And it is quite evident how we are performing way better for minor. And when it comes to major, they seem to show performance comparable to ours. And this is the overall. And as you can see, our method is performed significantly better than all of the methods. And finally, inter-dataset cell type annotation. So here evaluated on 26 trained test pairs across 4 experiments and 3 technologies, and we are at 74%, and the second best performing is 60%, which shows that our tool is doing pretty good in capturing cell identity genes and performing annotation. So yeah, in summary, we have developed a single unified framework for capturing cell identity genes with a robust cell type annotation module and interpretability capability, as well as capability to integrate diverse molecular modalities. So yeah, finally, I'd like to thank my supervisors, Pengyi Yang and Ellis Patrick, as well as my colleagues at Children's Medical Research Institute and Sydney Precision Data Science Center for their valuable feedback. Thank you very much.